Hello students. So we will be starting with the zoology optional, which can fetch you good marks if you have a proper strategy and proper way of writing the answers, well structured answers, and for that you need proper content too. So today we will be discussing the classification and relationship of various phyla up to the class level. So this is the first unit of your zoology optional. It includes some subtopics which are acoelomate and coelomate, protostomes, deuterostomes, bilateral, radiata, major minor phyla, status of protozoa, status of parazoa, then status of onychophora, hemichordata, symmetry, relation between symmetry and civilization. So these are some of the subtopics which are in the first unit. And when you have to prepare for this for the optional or any subject, the most important thing is syllabus and the previous year questions. So if you go through the previous year questions here, the previous year questions, the question here are related to recent system of animal classification, difference from earlier empirical approaches. Then some questions related to rotifera, which is in the minor phyla. Then there is question about Acoelomata. There are questions related to coelom, protostomes and deuterostomes, bilateria and radiata. Then also regarding the Parazoa, status of onychophora, and affinities of onychophora. So this is some of the general overview that what are the questions like, what from which portion the questions come. So all the portions, almost all the topics have been covered from the syllabus. So yes, you can expect questions can come from these topics in future too. So let's start by the first topic, that is the idea of classification. There is one history behind the development of classification over the years. The first type of classification that was given, it was the two kingdom classification, where the kingdoms, where the organisms, they were divided into two kingdoms. It was given by Carolus Linnaeus, and the two kingdoms were Plantae and Animalia. Plantae contained all the fungi, bacteria, algae, because it was very rudimentary and primitive type of classification. They did not have any idea that fungi can be kept separately or bacteria or algae can be kept separately. So they found the qualities, the outer external qualities, on the basis of them. Carolus Linnaeus simply divided into Plantae and Animalia. It was a simple classification, but it did not distinguish between unicellular, multicellular, or autotroph or heterotroph. So as time passed by, research was done. Then three kingdom classification was given by Ernest Haeckel in 1866. Now the kingdoms were divided into, now we had three kingdoms. It was Plantae, Animalia, and Protista. So now the unicellular organisms, they were kept in Protista. The unicellular organisms were kept in Protista. This was the three kingdom classification. As we move ahead, four kingdom classification was given in 1956 by Copeland. Now the organisms were divided into Plantae, Animalia, Protista, Monera. And Monera included the prokaryotic organisms. It included bacteria and cyanobacteria. So now the issue that was there in three kingdom classification, because Protista contained all the unicellular organisms, where prokaryotes as well as eukaryotes were kept under the Protista. So it was kept under the Protista. The limitation about the the limitation about the three kingdom classification, that is they were mixed there, they mixed the prokaryotes and eukaryotes, it was overcome in the four kingdom classification. Then we have five kingdom classification. The issue with four kingdom classification was that fungi was still in the plants, but the characteristic that were studied over the years, they found that plants and fungi, they have totally different characteristic. The internal systems are different, the anatomy is different, the way of reproduction is different. So over the years, what happened, R.H. Whittaker in 1969, he gave the five kingdom classification, that is Monera, Protista, fungi, plantae, animalia. Monera included the prokaryotes, Protista included the unicellular eukaryotes, fungi included heterotrophic saprotrophs, plantae included autotrophs, and then animalia, the multicellular heterotrophs, the advanced level of organism that was included in animalia. Then the good thing about this five kingdom classification was that it was on the basis of proper organization, body organization, cell type, nutrition and reproduction. But at the same time, it did not reflect the genetic relationships accurately. That is why in 1990, new kingdom of classification came, that was a sixth kingdom classification. And it was somehow one of the modified versions given by the Whittaker. Whittaker gave the five kingdom classification, and it was then modified, and it was also supported by Carl Weiss. In this system, there is Archaebacteria, Eubacteria, Protista, fungi, plantae, animalia. So the difference was here. Protista was there, fungi was there, plantae bhi tha, animalia bhi tha, but yes, Archaebacteria, the primitive type of bacteria, and Eubacteria, which had different lineages, they were formed. They were given separate kingdoms in sixth kingdom classification. So it had the first quality of differentiating between the true bacteria and the Archaebacteria. Then we have three domain system. Three domain system was proposed by Carl Weiss in 1990, and it is not a kingdom system but a higher level of classification. It contained domains, three main domains. First domain was archaea, second one was bacteria, third one was eukarya. And these were three domains, and these domains contained further multiple kingdoms. So this was something that was an advancement of whole classification over the years, and it was important because the organisms had to be classified in particular phyla, particular groups. So a proper hierarchy had to be made. Like here in archaea, there was Archaebacteria. In bacteria, we had Eubacteria. Then in eukarya, we had Protista, plants, fungi, animalia. So this is how the classification goes on. This is one of the general overview that how classification developed over the years. And yes, questions can be asked, the classical system of classification, new system of classification. So this is somehow the new system of classification. So if we see the recent system of animal classification, it is phylogenetic classification. It is based on evolutionary relationship. It is derived from common ancestry, not like the traditional system where the classification was done on the basis of outer structure. Outer structure. Old system was based on somehow on the basis of outer structure. Now the recent system of classification, it consists of cladistics, which include the clades. The clades are those groups of animals which share common characters, share derived characters. They have one clade contains some common characters, so they have the synapomorphies. These are the synapomorphies. Again, the recent system of classification, it has molecular taxonomy. Molecular taxonomy means it looks at the molecular level. Molecular level includes DNA, RNA, protein synthesis, sequencing, and on the basis of these molecular taxonomies, molecular structures, they are kept in one group. On the basis of computational tools are used over the years. You must have heard about the bioinformatics and softwares like Mega, Blast, ClustalW. These are some of the tools which are used to make these phylogenetic trees. You must have studied somewhere phylogenetic trees or seen. So these are made using the computational tools. Then three domain system is in the modern recent animal classification. As I said, it contains three main domains, and then there is further classification. And this three domain system, it is based on the ribosomal RNA sequencing. Again, important point that you will note down in the exam. Then it is one of the integrative taxonomy, as it combines morphological, molecular, ecological, and behavioral data for holistic classification. And this data is then used by these bioinformatic tools to create phylogenetic trees. So when we differentiate based on previous year questions, when we differentiate between the empirical classification and modern classification, the old one was simply based on morphology, anatomy. New one is based on genetic and molecular data. The approach earlier was artificial or natural, but the new one is phylogenetic and molecular. Then old one contained scientists like Aristotle, Linnaeus, but new one had Haeckel, Whittaker, Carl Weiss. There was distinction. There was very minimal distinction between the cell type, but now we have clear distinction between the cell types, whether it's prokaryotes, eukaryotes, and earlier the tools were only based on the visible trait because the technology was not much advanced due to lack of technology. But today, the sequencing, we have different bioinformatic tools. We have molecular phylogenetics, we have DNA and RNA sequencing, we have biotechnology, and on the basis of this system of technology, there has been advancement in classification too. Then the evolution was not considered at that time. The evolution was not considered, but now evolution is considered as a central principle of the classification. Old one contained systems like 2 or 3 kingdom, and now we have 5 kingdom, 6 kingdom, 3 domains. So you have to write these in exam. If and then the accuracy, earlier it was less accurate and now it's more accurate. Earlier it was often arbitrary as fungi and plants were kept in one group, but now it is based on common ancestry and molecular similarities. So this is something that we covered and it had come already in the previous year questions. So If you will look closely, okay, what comes under the classification, it is actually a very systematic method to categorize and organize biodiversity based on shared characters. So whenever the question comes, you can introduce classification as systematic method to categorize and organize the biodiversity based on shared characters. If you will see, if we have plants, and plants, these have leaves, branches, chlorophyll. So organisms which have these minimum or maximum shared characters, they are kept in one group. This is how the classification is done. So we classify them in on the basis of shared characteristic. And there is one hierarchy of classification. You can remember this mnemonic or this sentence ki, dear King Philip came over for good soup. So dear King Philip came over for good soup. Dear is domain. Domain is the highest level. And then in domain we include kingdoms. Kingdoms. After kingdoms we have phylum. After phylum we have class. After class we have order, then family, then genus, then species. So domain is the largest and most inclusive category. For example, we saw eukarya. Let's take human as example. In kingdom, we have groups of organisms broadly containing, for example, we have Animalia, we have Plantae. So kingdom includes Animalia, Plantae, Monera, Protista. Then we have phylum. Phylum, it is based on the body plan and organization. Now, in the syllabus, we will be discussing many phyla. Though those will be phyla. In the phylum, you will see there is proper body plan, proper body organization. And in case of humans, the phylum is Chordata. Then in case of class, it's Mammalia. Mammalia is further division within the phylum, and these classes have shared characteristic at the level that is below the phylum. Further, they have been divided into classes. Then the classes have been divided into order. Order में आएंगे families. They are the group of families where they have similar traits like primates. Then family contains more closer shared features like hominidity, humans. Then after that we have genus. In genus we have very closely related species. Homo, and species is the basic unit of classification, Homo sapiens. So I hope this is somewhat understandable at this point of time. And if you will see, in case of house cat, Eukarya, Animalia, Chordata, Mammalia, Carnivora. Now this Carnivora, it contains different families, and in case of cat the family is Felidae, and in case of genus it's Felis. So you understand the concept of family here. The family contains group which shares very common features among them. After that, the domains. Domains I already explained earlier. Archaea was there, bacteria was there, eukarya. The simple, the characteristic of domain was that in archaea we had prokaryotic, unicellular organism. Cell wall lacks peptidoglycan. It is one of the component of the cell wall, but it's not present in archaea. These archaea live in extreme environments. These are thermophiles, halophiles, and they have distinct genes, metabolic pathways from bacteria. And then bacteria, it's prokaryotic, unicellular. Cell walls contain the peptidoglycan. Here is the difference, and it's found in diverse environments. They include both the beneficial as well as pathogenic species like E. coli, streptococcus. Then eukarya contains eukaryotes. Including the unicellular or multicellular, and these include the four kingdom: Protista, fungi, Plantae, Animalia, like humans, plants, amoeba, fungi. So this was about the general overview about the domain and classification. Everything about classification has been covered. Now if you go through the system of level of organization, this is level of organization. In case of level of organization, you can expect one question related to level of organization. If you see here, the Animalia have been divided into cellular level and tissue organ system level. In cellular level, you will observe there is one phylum, that is Porifera. We will be studying this Porifera in detail later on, and this Porifera, it is related to the sponges, and rest we have tissue organ or organ system level of organization, and it is observed in rest of the phyla. So level of organization is actually the hierarchy of structure within the living organisms. It is a level of hierarchy within the living organisms. So the level of organization that is shown by the organisms, it is cellular. Then as we move ahead, it is tissue level. There is organ. Then we have organ system, and then you will observe the organism. In case of cellular, you will observe that it is the cells are the functional unit. You will see one cell. For example, you have amoeba.